the kind invitation and for everyone for logging in. I'm really glad to have this opportunity to speak at the Lingjun webinars series. As my title suggested, I'm a professor from University of Wisconsin-Madison in School of Pharmacy and Department of Chemistry. I'm really glad to have this opportunity to talk to all of you about some of our effort on developing chemical tags to facilitate quantitative glycomics and glycoproteomics. I guess for most of the audience on the call, probably it's not so necessary to really emphasize the importance of glycosylation and their importance for functionality of proteins. So glycosylation is arguably probably one of the most diverse and complex post-translational modifications on the proteins. So not only they actually come with multiple forms, but not only it will change the structure and conformation of these proteins, and also it will change actually the functionality of many of the glycosylated proteins in impacting cell-cell recognition, intercellular communication, and immune response. And consequently, there are actually many literature report aberrant expression of glycosylation is implicated in various diseases, including multiple types of cancers, cardiovascular diseases, and also neurological disorders. Consequently, it's important to quantify glycans with high accuracy and precision, and also in high throughput. So in the mass spectrometry field, there are a number of ways that you can quantify glycomics, glycans, and glycopeptides. I saw Graham is on the call, and he wrote this very nice article to kind of summarize the current strategies and approaches to quantify glycans. So we can do this on the freely released glycan level or intact glycopeptide level. And this can come with different versions, starting with the simplest label-free approach or metabolic labeling. Alternatively, you can also turn to chemical tags, and this comes with different versions, such as MS1-based quantitation using isotope labeling to introduce mass difference for example, the light and heavy version of samples to relatively quantify, or if you want to do higher throughput methodology that we can refer to isobaric labeling, and the quantitation occur at the second stage or tandem mass spec fragmentation. And this can also come with data dependent or data independent acquisition mode. So among these different quantitation approaches, there are pros and cons. For example, metabolic labeling can be very accurate. However, it's also quite expensive. Stable isotope labeling for the MS1-based approach oftentimes a problem to introduce spectral complexity and also isotope overlapping issue. And there's also a limitation usually on the plexing that you can go. So mostly duplex or triplex quantitation. So in contrast, MS2-based quantitation such as amnioxy tandem mass tag or TMT can offer higher plexing, higher throughput approach. However, it still has the limitation sometimes labeling complex glycans and also produce lower reporter ion yield. So to address some of these limitations, our lab has been developing several chemical tags. So I will introduce the first one is the MS1-based quantitation dimethylpyrimidine, also known as dipyrotags for glycan quantification. So shown here is the structure of the dipyrotag. So we have a amine reactive group and also this pyrimidine group and a dimethyl group where we can incorporate, for example, different heavy isotopes. So here shows the different configurations that enable this dipyrotags can allow duplex, triplex, all the way through aplex quantification. So this tags was originally developed for quantitative, actually quantitative proteomics and peptidomics. And some of the features I want to highlight. So this is a relatively compact chemical tag. So we only add about 254 Dalton upon labeling. And also, as you can see, we utilize the mass defect labeling approach. So that would introduce very small mass difference between adjacent quantitation channels. so that in the low resolution mode, these peaks actually, these MS1 quantitation does not introduce spectral complexity. However, when we use higher resolution instrumentation, here we can actually allows us to separate or quantify 2 or 3 samples and all the way to 6-plex. And this plot actually highlight the quantitation performance as a function of the Orbitrap resolving power. So again, as you can see, the higher resolving power actually offers a better quantification that allows more sample to quantify. And I want to also add this compared to MS2-based approach, this overcomes some of the problem that with the MS2 isobaric tagging, that is the co-isolation interference. So obviously we like to extend the utility of this dipyro tag to glycan quantification. So to do that, we actually take the glycoproteins and release them, release the glycan with pangase F, and under the alkaline condition, this would generate glycosylamine. And this amine group can then react with the dipyro tag for quantitation. So here we can actually achieve the synthesis of these dipyro tags in actually 3 simple steps. So we started out with arginine, with formaldehyde reaction to dimethylate this group, and then we can convert the guanidinium group to pyrimidine group using the acetone. And then we can convert the carboxylic acid, activate them to triazine ester to react with amine group. So shown here is a 2-plex dipyro tags where we can highlight in this case the red dots showing the light version of dipyro tags where the heavy isotopes are N15 or O18, and then the blue dots signifying the heavy dipyro tags where we incorporate 6 deuterium atoms. So the first thing you might be wondering, because these 2 tags actually have very small mass difference, what kind of mass resolving power that is needed? So here we actually systematically evaluated a number of different resolution that is required to separate these dipyro tag labeled neutral glycans. As you can see here on the top, 120,000 resolving power is required to separate actually the light and heavy of the H3N3 glycans. And the bottom for larger glycans, here we require 240,000 or up to 500 K resolving power. So then we want to incorporate this to our workflow to look at glycoprotein release glycans with the dipyro tags. So here we're starting out with a glycoprotein denature and then pangase release and differentially label these 2, the glycans, 2 samples. And then as we can see, in this case, using resolving power 500K, we can readily resolve these 2 light and heavy dipyro tags that is 45 millidalton apart from each other. So shown here is an example of looking at a particular glycan labeled with light and heavy dipyro tags. And with the high resolving power, we can see that we can separate these labeled in 1-to-1 ratio. And the area under curve, the extracted ion per monogram actually shows the overlapping. So that suggests we can actually utilize this to accurately quantify these glycans. And the bottom shows the fragmentation spectra where we can actually identify these glycan very readily. So then we want to apply this to look at a more complex sample. In this case, we're applying the dipyro tag to perform relative quantification of glycans released from the human serum protein. This was collected from patients that treated with chemotherapy or the leukemia. And so here, this is actually one of the most predominant childhood cancers. And there's a lot of concern about the neurotoxicity and looking at the CSF in serum samples. So using our dipyro tags, we can actually quantify the glycan, number of these glycan expressions before and after chemotherapy. As we can see, the majority of these glycans actually show downregulation. In particular, we highlighted 4 of these glycans, both the neutral glycans and also the sialic acid containing glycans. So this just kind of summarize this part. We basically use previously developed proteomics tags, the tag, that allows us to actually perform relative quantitation for glycomics and glycan quantification that using high resolution mass spectrometer. And so this actually also unlocked the potential for use some of the previous developed proteomics tags for quantifying glycans. However, there are still some limitations that you can probably recognize. One is that this type of tags, like MS1-based quantitation, really rely on high resolving power instrument. In fact, if we want to go higher plexing, this would require ultra high resolving power instrument, which may not always be readily available for many of the labs. So to combat that problem, we turn to isobaric tag labeling. This would allows us to interrogate many more samples. So one of the more successful commercially available tag is the amnioxy or the glycoproteomics tag, or TMT. So shown here is a generic glycan amino-oxy TMT tag structures. And so, and this would, the amnioxy group would react with the carbonyl group, or the reducing end of glycans. And we also have a mass balancer and a reporter. So this reagent is configured in a way where we can have the reporter line from 126 to 131, one dot apart from each other, and the balance group. So the reporter and the balance add together would be identical mass. So in this configuration, we can differentially label 6 sets of samples and combine them in the mass spectrometer. So in MS1, each of these glycan from 6 samples will show as a single peak. There's no spectral complexity introduced. And then upon tandem mass spec fragmentation, this would generate reporter line that allows us to perform relative quantification and the backbone fragmentation for identification. So showing here is the commercially available 6-plex amnioxy TMT, where the red asterisk suggested the position of the stable isotopes. Again, this is actually aldehyde or ketone reactive group react with the carbonyl group to form this hydrosome, and then in CID mode to cleave this bond to generate the cyclic reporter ion. So this approach has been very actually popular. However, when we apply this to our complex glycan quantification, we noted a few problems. One is that this actually caused incomplete labeling for many of the complex N glycans. And also there is actually a production of the reporter ion yield tended to be low. And when we use the low collisional energy, and this would cause a problem for inaccurate quantitation. And because we want to get the backbone fragmentation. When we raise the collisional energy, this would generate robust reporter ion that give more accurate quantitation. However, as you can see, there's no backbone fragmentation. So in other words, we have to combine or use 2 different collisional energy to get both quantitation and identification information. So to combat this problem, again, we decided to actually develop our own custom designed tags. So here we turn to our previous designed dimethylated leucine tag, which has been extensively used for quantitative proteomics and peptidomics. So here shows the structure of our leucine tag and react with formaldehyde to form this dimethyl group in reducing reagent, and then react with a glycine methyl ester to install the glycine linker. And this would then react with hydrazine, the hydrazide group that is highly reactive to reducing end of glycans, in particular, actually all the carbonyl containing compound. So this would allow us to basically, in this case, by installing different stable isotopes, such as color-coded here, C13, deuterium, and M15, we can create a set of 4-plex sugar tags that is 1 dalton apart from each other for the reporter ion. So then based on this idea, we can actually design a 4-plex quantitation schemes in looking at a glycans released from a glycoprotein bovine thyroglobulin. So again, we started out with the natured protein released with pangase F for the N glycans. And here we can aliquot them in 4 different ratios and label them with our 4-plex sugar tag, and the MS1 mode that we to perform the full scan, and then MS2 that allows us to generate the reporter ion for fragmentation. So obviously the first thing we want to do is to compare our tag with the commercial tag to see the labeling efficiency. So here we can see that the unlabeled or the native glycans are shown here, and the amnioxy-TMT gives rise to actually about, in this case, 50% of these still unlabeled. And in contrast, our custom designed sugar tag enabled full, nearly 100% conversion with labeling efficiency. Another very important factor that we need to compare is the MS2-based fragmentation. As here we compare 2 different HCD energy level, the 25 and 30. And on the left is the amnioxy-TMT labeled with the neutral N glycans. As you can see, in both cases, the sugar tag labeled produce more intense reporter ion for better quantification, and also the backbone fragmentation allows us to find the glycan structure. And so I want to point out this more robust intense reporter ion means in general we can quantify glycans with higher accuracy, and also allows us to look at lower abundance glycans, which is obviously important. And this is another example to show that our sugar tag also works well for fucose and sialic acid containing glycans. Again, compared to the amnioxy-TMT, this shows about 4.5-fold increase in the reporter ion production. So then we want to apply this to look at a more complex glycan labeled with 1:1 to 5:10 ratio. So showing on the top is a box plot to look at the quantitation performance, and the bottom are 2 representative glycans. Again, we can label and quantify them in pretty good accuracy, 1:1, 5:10 ratio. So next we want to again apply this to look at a more complex sample. So we turn back to this serum samples collected from children that receiving the chemotherapy who suffered from B-cell acute lymphoblastic leukemia. And in this case, we're looking at 4 different time points. So basically before the chemotherapy, in 1 month, 3 months, and 6 months after chemotherapy to look at the dynamic change of the glycans. So here we can actually pair this with our 4-plex sugar tag to look at these 4 different samples, and showing on the bottom are the results to compare the chemotherapy treated versus the control. As you can see, we can altogether quantify 145 N glycans, and among these, 68 of these N glycans are commonly shared in all 3 patients. So now we can actually do this in 4-plex version. So we want to further improve the throughput to enable us to look at a larger number of samples. So there are a number of advantages of doing so. One is that it will help us to reduce the sample amount from any one sample, and also it will reduce the sample preparation and analysis time. And perhaps more importantly, this would allow us to look at many more biological conditions or replicates in a single experiment. And also it will increase the LC-MS data overlap. So to achieve from 4-plex to 12-plex, again, we turn to this phenomenon called mass defect. And based on the Einstein's energy equation, E equals mc squared, the difference in the neutron binding energies actually result in a very small difference in mass. For example, if we swap 2 of these deuterium atoms to carbon-13, this would give rise to 5.84 millidalton mass difference. And that can be actually resolved with high resolution instrumentation. So we actually kind of systematically evaluated different reporter ion and the required mass resolving power. So based on this idea, we have designed and synthesized a 12-plex version of the sugar tag. So this would represent threefold of enhancement in the throughput and multiplexing capacity compared to our 4-plex, and showing first of these 12-plex tag. Then also in this MS2 mode that we can generate this reporter ion. And this work was done by two talented graduate students, Yufeng and Mike Li, and both recently graduated. So here we can see that first thing we want to look at is to what kind of resolution that is needed to separate these 12 quantification channels. As you can see from this plot, that actually as low as 30,000, we can already actually separate these reporter ion pretty readily. Obviously, the higher resolving power would allow us to actually separate these 12 channels even better. So then we want to also evaluate their chromatography behavior. So we took 5 of the representative quantitation channels and run them on the LC-MS, and as you can see, they have actually pretty good consistent retention time. So that will allow us to do more accurate quantification. And the box plot also shows that they actually shows reasonable good quantitation performance. So then we want to apply this 12-plex sugar tag to quantify enzyme release from human IgG. So there are 4 different isotypes of IgG. In particular, we want to, and there is a lot of interest these days to look at these therapeutic monoclonal antibodies, or in the context of antibody drug conjugate, to characterize the specific glycan expression patterns from these IgG isotypes. So what you can see here, we actually took 4 of the most abundant N glycans and compare their relative ratio or expression levels. And so, for example, here for the G0F or the G2F, and we can see there is actually opposite changing trend. In the other, the G1F, the 2 of these structural isomers, that each of these IgG isotypes have its unique relative expression levels. And here in the bottom is one example where we can actually quantify 12 samples. So this is 4 different isotopes in 3 biological triplicates. So that allows us to, in a single LC-MS run, to quantify all these glycan expression levels. We want to also try a little even more complex samples. In this case, we are applying the 12-plex sugar tag to look at the ECC1 cell treated with the atovaquone, which is an antimicrobial medication that is used to treat pneumonia and malaria. And it's also an inhibitor of the dihydroorotate dehydrogenase, and it can inhibit pyrimidine synthesis and UDP glutamate generation, which leads to decreased O-glutamylation. So we reasoned that this could also change the N-glycan expression upon different treatment. And so here we took 2 different time course, 48 hours and 72 hours in these 4 samples in biological triplicates. So here shows the several of the representative N-glycan expression levels. So altogether, we were able to look at 55 N-glycans and quantify 43 glycans that express in all 12 samples. So, so far I've talked to you mostly about the freely released glycans. And next I want to spend a little bit time to talk to you about how to use chemical tags to quantify intact glycopeptides, in particular under disease in normal conditions. And the applications that I want to highlight is the Alzheimer's disease biomarker discovery. And for people on the call, probably everybody's quite familiar with Alzheimer's disease, and this is one of the most common dementia, and currently affect more than 6 million Americans that are currently living with this devastating condition. And 1 in 3 seniors actually die with AD or related dementia. And this number was actually taken from a recent stats number in 2022. Alzheimer's and related dementia actually cost our nation over $321 billion US. And this number is going to rise to nearly 1 trillion by 2050 if we don't do anything. So clearly developing strategy or diagnostic tests at early stage would help to alleviate some of this problem. So this work was done in collaboration with two fantastic clinicians, Cindy Carlson and Ozima Onkoko, both from Wisconsin Alzheimer's Disease Research Center. So as many of you know, that Alzheimer's disease is a progressive neurodegeneration that can be divided into multiple phases, from preclinical to mild cognitive impairment to dementia, based on their neuroanatomy factors or physiological and more on biochemical signatures. And many of you familiar with some of the protein-based biomarkers such as amyloid beta, tau, or phospho-tau. But we believe that glycosylation could really play a very important role based on some of the literature suggesting that these glycosylation pattern changes that play an important role in initiation, progression of AD. However, currently there's actually no systematic or comprehensive evaluation of glycoproteome in the, for example, the blood or CSF samples. So when we talk about protein glycosylation quantification, it can come with multiple levels. One is that we can look at the changes in protein abundance and highlighted in this blue circles, or second level is to look at glycosylation site occupancy, or oftentimes referred as macro heterogeneity. So for example, these protein B that highlighted in green triangle. So for example, we have a reduction of the high mannose structures. And the third aspect or situation is to look at glycoform changes at specific site, which is oftentimes referred to as microheterogeneity. For example, here, the purple protein C, in the disease condition, we can see there's sialic acid being expressed or upregulated. So over the time, our lab has developed an integrated analytical platform to look at glycoproteome expression in cerebrospinal fluid. And again, this was done in collaboration with Wisconsin Alzheimer's Disease Research Center. So we took this CSF sample, extract proteins after enzymatic digestion, enrichment, and separation, and then with a variety of different fragmentation techniques to perform site-specific quantification, characterization, and then we can use a variety of ways, in particular isotopic isobaric labeling for quantitation. So with this integrated approach, we were able to achieve nearly 30-fold enhancement for N-glycopeptide characterization so over nearly 3,600 N-glycopeptide in the cerebrospinal fluid, and this also mapped to nearly 360 N-glycoproteins. That's a 10-fold improvement. So first thing we want to look at is to actually look at a global N-glycoproteome expression level in the healthy control and AD. As you can imagine, probably majority of these proteins are shared. So here shows the N-glycoproteins and N-glycoside that is commonly shared between the control and AD. There are still some unique expression levels. And also we can actually see that if we look at broadly 6 different classes of these different glycans, such as high mannose or sialic acid or fucosylation, or the combined sialic acid and fucosylation. So the first thing we want to do is to perform this hierarchical clustering to get a bird's eye view of the number of detected glycoforms in the AD sample versus control, and here visualized in this heat map. So among the 373 shared N-glycans, we have about 1/3 that don't show changes, and 2/3 of these actually show substantial changes. And again, we categorize them in these 6 different classes. And as we can see that pretty readily, you can see actually a substantial reduction of the fucosylation. So the number of glycoform changes in AD versus control, and color-coded here, the blue suggesting downregulation. And so we can see actually quite a few downregulation of the fucose, broadly speaking, and also some of these fucose and sialic acid combined. So then we want to also look at a little bit closer the microheterogeneity of some of these proteins. So one of the example highlighted here is the alpha-1 anti-chymotrypsin. So this is a glycoprotein involved in amyloid plaque formation. And we can see there are 4 different N-glycosides in the control. We can see, for example, the 106 in the 80 sample, we actually look at substantial upregulation in, for example, by the way, the blue circle suggested a sialic acid, so there's a different color coding. And here we can also see different changes in, for example, one of these N-glycoside does not have glycosylation. And another interesting glycoprotein is the afrin A3. So in the control sample, there is a single asparagine site 38 have 5 different glycoforms. And in the 80 sample, there's actually no glycosylation detected. And the afrin EPH signaling pathways is involved in neurotransmission. So the dysregulation of the glycosylation in the 80 sample, again, may implicate the impairment of the neurotransmission. And another very interesting glycoprotein is the hanasinase CN1. So here this shows a single asparagine site, 382. Again, in the control sample, we actually observe 19 of these different glycoforms. And the 80 sample is actually a substantial downregulation. And this is also an interesting dipeptidase that cleaves between the beta-alanine and histidine. So in normal aging patients, there is actually increased carnosinase secretion in the CSF sample. So again, the N-glycosylation is actually related to the secretion. So the reduction of these the N-glycosylation in 80 sample could also be responsible or contribute to the reduced secretion. So again, that could be another interesting biomarker. So in order to further improve the throughput for quantitation and also increase the number of samples that we can interrogate, we again turn to high throughput multiplex tagging strategy. So here we again use our dimethylated leucine tag. So this is the trisine ester to react with primary amine, the carbonyl group, and the dimethylated group, where we can install different stable isotopes. So using mass effect, we can actually create this 12-plex dilute tag. So we can then use this to simultaneously interrogate, in this case, 48 patients that divided into 3 groups: the control, mild cognitive impairment, and advanced dementia. So 16 patients in each group. And then we can perform this analytical workflow, use our 12-plex dilute to quantify them. So this allows quantifying over 1,500 N-glycopeptides, and that map to over 107 glycoproteins. So then we want to look at the glycosylation patterns. In particular, we also noted there is a majority of these glycosylations happening in the fucose or sialic acid or the combined modification. And when we look at this pie chart, it's noted that more than 60% of glycoproteins actually has only one glycoside. However, more than 70% of these glycosides have multiple glycans. And so this glycoprotein network map, so actually we can utilize this kind of a wheel plot to understand better about the glycan heterogeneity. So the outer node, there's 157 glycans, and we can map how they modulate and modify the 107 glycoproteins that organize in this inner bar based on the sites per protein. So again, the majority of these modifications happen from sialic acid glycosylation or the combined modification. So another way to look at the glycan heterogeneity is to plot the number of glycans as a function of number of glycosites. So for example, the contactin one that has actually 15% of sialic acid, but there's 30% of high mannose. In contrast, the ACT one have 50% sialic acid, but no high mannose modification. So we also take a closer look at the microheterogeneity of these glycoproteins. So I highlighted 3 examples here. One is the neural cell adhesion molecule, L1-like proteins. There are 4 different glycosides, and there's moderate degree, a low degree of glycan heterogeneity. And complement C3 is a neuroinflammatory protein. There's only one glycoside at 85 asparagine, but there are 22 different glycoforms that we can identify. And clustering is really interesting. So that's the third most risk gene in AD, and it encodes 449 amino acid residues. So there are 4 different glycosides. Some of these have very low degree of heterogeneity. However, at 374, there are actually 56 different N-glycoforms, and many of these are fucose and sialic acid modified. So at the global level, we've also looked at the dysregulated N-glycopeptides in the MCI and AD. So we can see there are 69 and 132 of these N-glycans showing dysregulation. And the gene ontology analysis suggests the top-ranked biological processes include platelet degranulation, cell adhesion, and also response, and many of these related to neurodegeneration. So we then took the 19 of these N-glycopeptides that showed consistent changing trend in the MCI and AD, and trying to figure out whether the change happened at the PTM level or at the protein level. So to answer that question, we performed in parallel the quantitative proteome analysis using the 12-plex dilute tag. So one of the examples highlighted here is the apolipoprotein D. As you can see at the protein level, there's no substantial changes at the glycopeptide level. So for example, here at 65 N-glycan, we actually show 8 of these N-glycopeptides show upregulation in both the MCI and AD, and the 98 position that actually show opposite changing trend. And that's because that this particular one is actually occupying the coil junction that connecting 2 of the beta strands. So that suggests some structural implication. But the final piece of data that I want to show is that we can also use this approach to look at disease stage specific N-glycopeptide changes. For example, one of the glycoprotein prostaglandin H2D isomerase, we can see there are 2 of these N-glycoside at 51. Both the MCI and the AD shows downregulation. And at the position of 78, at the MCI, there's an upregulation. And then at the AD condition, there's substantial downregulation for all of these different glycoforms. And there are also a number of these other interesting changing forms. Due to the interest of time, I'm not going to go through it. But the final kind of a ongoing point that I want to briefly highlight that we're pursuing is to further improve the sensitivity of this dilute tagging strategy, where we can incorporate a boost channel. And the general idea is that because of these quantification is based on tandem mass or MS2-based fragmentation. And so for low abundance species, they won't get selected for MS2. So in order to overcome this issue, we want to introduce, for example, and select one of the channel with a high abundance or a large quantity of samples. In this case, we can select, for example, dilute 118D as a boosting channel. So here we can actually adjust the ratio of the boosting channel and the study channel, for example, in this case, is 30-fold more. And then the boosting channel is basically result from pooled CSF samples from both AD patient and non-AD patients. So that will have actually very similar biological matrices. So this would ensure that we can actually introduce, for example, the MS to generate the reporter line that allows us to quantify things. So this is an ongoing effort. We can use this to improve the sensitivity. And so with the boosting strategy, we were able to quantify over 1,172 N-glycopeptides that map to 164 glycoproteins. And this also revealed near 50% of the glycopeptides were silylated, and 22% of these are fucosylated. And so we were able to both look at the glycoproteome globally, but also looking at, for example, 18 of these glycopeptides are dysregulated, 9 showing upregulation, and 9 show downregulation, and 72 of these dysregulated glycopeptides were silylated and 38 were glycosylated. And so this would basically allow us to look at things with some of these more sample limited situation. So to kind of wrap up, I hope I've shown you today that we were able to develop several chemical tags that one kind is related to MS1-based quantification using dipyro tag, and the second stage MS2 level quantification with the sugar tag to allow us to do multiplex quantitation for glycan expression. Also, we have cost-effective isobaric dilute tags that enable quantitative glycoproteomic analysis in biofluid samples for biomarker discovery. Our current ongoing strategy is to incorporate boosting strategy to further improve the sensitivity of looking at these intact glycopeptides from limited samples. So with that, I want to thank the graduate student who contributed the work. I highlighted their names here, and also collaboration from Cindy and Ozima from the Wisconsin ADRC, and special thanks for the NIH Glycoscience Common